Presenting here. Hello, can you hear me well? Thank you. Okay, first of all, I want to thank the organizing committee. It's an honor for me to be here, surrounded of very big names, and I'm afraid I have to do a very good job, so you won't be disappointed with my presentation. And today, okay, I'm gonna talk about CHiDO. I don't know how many people here speak Spanish, but in Mexico, CHiDO is a very interesting phrase that we use to describe something good. So, for example, if somebody asks you, how do you like living in College Station? If you like a lot, you can just say CHiDO. So we developed this implementation to simplify the data analysis and stream of multi-omic data. So how we can integrate multi-omic data in a very easy framework. So this is the outline of the presentation. First, I'm going to talk a couple of topics about science, so deep learning optimization. And we will describe a few of the things that we found now that everybody is jumping in the area of AI. So we wanted to analyze or to study in a better way what's going on with the architectures. And then I'm going to mention about sparse tested design, how we can apply this to the industrial level, how we can optimize the allocation of resources, and how we can get products faster. And then I'm going to talk about CHiDO, so the platform, what it is, how we can access to the platform, and what type of analysis we can conduct in this case, right? We can use for genomic prediction, we can use for hybrid prediction when you have genomic information from the parents, or if you are trying to study stability patterns. So let's start with the deep learning optimization. So as we know, several studies have been presented mentioning the use of deep learning in genomic selection. So some of these have shown moderate improvements, but in general, we don't see that significant improvements with respect to the conventional tools that we have, like G-block model, for example. So we know that the AI is increasing our expectations to improve, significantly improve the results compared with the conventional tools. So one of the problems is most of the methods that we are using, we are trying to adopt in genomic selection are methods that were developed for other type of problems, like chatbots, healthcare engineering, where we have the same type of data. So image recognition, everything that we have are images. In our case, in plant breeding, we know that there are several factors affecting plant performance. It's not just about genetics, it's weather, management, etc., anything that you can put on top of the implementation. And the implementation works, right? But the implementation many times is tricking the user, right? The implementation is intelligent, finds what is the layer that optimize or reduce the noise or a loss function between predicted and observed values and just focus in that specific layer. In a way that it could be risky for a breeder to give a recommendation for a specific set of weather conditions, right? If you are a true breeder, you know the most important factor that drives variability for you should be genetics, not the weather, and you can make wrong recommendations if you don't pay attention on this. So we know that using deep learning is a little tricky because it depends on multiple factors, what we call the architecture, right, or the layers. So we have number of hidden layers, activation functions, neurons per hidden layer. So we might have different number of neurons for each one of the different layers, learning rate, number of epochs, batch size, dropout, etc., etc. So as you can imagine, we have many factors that we should optimize. So to our knowledge, and I might be wrong, right? I'm not saying that I read a lot. So node optimization has been conducted to select the best set or to find the best strategy of hyperparameters to apply in genomic selection. So also we had the genomic selection depends on many different factors. obviously genomic information, the quality, LD, trait architecture, and quality of the phenotypes, right? So in this study, we attempt to find these patterns, right, in a way that we can provide a recommendation. They should be broad, not just find the specific combination that's going to give you the highest predictability, but what are the patterns that we should be improving or we should be modifying in order to accomplish a method that outperforms conventional genomic selection methods. So this is a study that corresponds to the master thesis of my student Ravendra Garapati. And in this case, we consider what I say a very comprehensive number of parameters and levels. As you can see here, it's not that comprehensive because we wanted to study more, but we have a lot of limitations. So we have 3 levels for dropout, 0, 0.25, 0.4, learning rate, 0.1, 2, and 3, lambda, right, the regularization parameter, 2 levels, neurons per layer, so each one of the layers, we vary the number of neurons, 3, 4, 5 for each one. Activation functions, we vary the activation functions at different hidden layers, so we consider 3 activation functions, so we have a total of 9 different combinations. And batch size 50 and 100, epochs 1000, 3000, and we have inner replicates. So we know that one of the downside of using deep learning is every time that we train the model, we select a random sample. So we repeat that random sampling 10 times to have better results. And we have a total number of 5,248,000 combinations. So we wanted to have more, but as you can see here, the factor that you multiply increases a lot the number of combinations. Also, we consider different cross-validation designs, and we apply this to a nested association mapping population in soybean. So we select a random sample of 1,379 genotypes that were scored in 6 environments and scored for 3 traits, grain yield, oil, and protein content. And we consider 2 different cross-validation schemes. CV1 that corresponds to the prediction problem of predicting the performance of genotypes that has not been observed in any other environments, and CV2 that corresponds to the incomplete field trial. So sometimes at the end of the breeding pipeline, you might want to test genotypes in multiple environments, but you might not have enough seed to conduct all of the testings. So this is the CV design. We have 2 cross-validations, 3 traits. We have 3 replicates, because imagine we have 5 million combinations, and we had 5 faults for each one of the different cross-validations. And we have to contrast with something. So we initially thought of using the G-blot model, right? The simplest method that we can use for running the comparison. And also the G/E reaction norm model, where we introduce in the model the interaction effect. And these were the results. So first, since we have a very large number of combinations, we wanted to find a way to simplify, to find patterns, how we can order the results in a way that we can visualize trends, right? So we did something very naive. We used regress, right, the correlation that we obtained for each one of the different combinations versus the mean and activation function combination. We have 9 combinations. So we have 3 different activation functions, sigmoid, relu, and tangent, and number of epochs, batch size, number of nodes in hidden layer 1, hidden layer 2, lambda, regularization parameter, dropout, learning rate, and error. So very simple linear regression. And what we found is that the most important or crucial factor explaining correlation were first, the activation function combinations, right? The second was the learning rate, and the third one, the number of epochs. Obviously, we have more factors that we study with more detail later. And this is how the result looks like. So we order first, we have 8 vertical orange bars that separate the results depending on the different combination of activation functions. So whatever is in the left side that includes sigmoid activation functions, either in the first or the second in both hidden layers, returned bad results. In the right side, we have those cases where the tangent activation function was combined, right? Either with ReLU or with the tangent activation function. So from here, these are the results for CV1, right? Predicting new genotypes. We can discard some of the activation functions that we don't have to look at. So for CV2, we have a similar trend, right? It's clear that we can see some patterns. Now we can identify some patterns that seem like as we move to the left side, the right side, we improve predictability. Also, we impose or we draw other vertical lines in blue. We have different learning rates, and seems like as we increase the learning rate, we improve predictability up to a certain point. And also within each learning rate, we have the batch size. In this case, we have 1,000, 3,000, so the quality of the image is not really good, but what we're observing there is as we increase the number of epochs, we improve predictability. So with this, we can use focus in the last 3 activation functions, or combination of activation functions, right? So here we use discard whatever was involves sigmoid, and then we pick only those that have the tangent embedded. So we consider only learning rates of 2 and 3, and then now we focus on what's happened with the batch size. So, and we have results like this. So now, still, we have a lot of results to condense, but now we present those in a box plot. So now here we have the 3 combination of activation functions, the learning rate, 2 and 3, and then we have the batch size. We have 100 to the left and 50 to the right, within each one of the blue lines. And then we have the number of neurons in the second hidden layer. So for us, what's interesting that it doesn't matter how many neurons you have in the first hidden layer. What matters is how many neurons you have in the second hidden layer. It's clear that as we increase the number of neurons in the second hidden layer, we can keep improving predictability. Probably the idea is to evaluate what happens if we increase the number of neurons in the second hidden layer. So as I said before, in the box plot, still we have many results, right? We have the results on 10 inner validations, 5-fold, across 6 environments, and the dropout levels and regularization parameters are confounded, but at least are not that crucial to affect predictability. So now we can focus on increasing the number of hidden layers without bothering about the other factors, because those are not crucial. So this result is for CV1, and something interesting that we found is that a low batch size returns higher predictability. So when the batch size was 50, we had better results than the batch size was 100, right? We don't know. And again, we increase the number of neurons in the second layer, we keep improving predictability, and we observe something similar for the case of cross-validation number 2, right, in complete field trials. So now we can provide a recommendation. So we can pick any of these box plot that return the highest predictability, and then we can suggest to the user to use that combination between activation function, batch size, epochs, etc., without considering that much other factors, right? So we contrast this with parametric methods, and we consider three models. Well, initially 2 models, a model without marker information, a model that includes main effects of the markers, and in this case, deep learning mean BC corresponds to the box plot that returns the highest correlation. And it's clear we can improve results with respect to the main effects models between 17 to 18% for grain yield for CV1 and CV2, but not for the other traits, probably because those are less complex. Anyway, we were curious and we wanted to contrast against the model that includes the GY interaction, and we found that although deep learning outperformed main effects model, it was no better than reaction norm model. In any case, we look for the highest correlation for any of the combinations using deep learning. And we found that the results were close to the reaction norm model, right? We have 53 versus 54, 53 versus 57 for yield. So probably if we keep increasing the number of neurons in the second hidden layer, we can reach or surpass the results from the GY model. Okay. So the conclusions, we had the deep learning is a powerful alternative dealing with large data sets, so we are happy for that. However, it requires an optimization process to find the best set of hyperparameters. And that's the downside, because it can be computationally demanding, right? So again, our results show that the number of neurons in the first hidden layer is not important, but in the second hidden layer, so probably we can keep doing studies on that. And then the learning rate saturates up to a certain point. After learning rate is greater than 2, we don't see any significant improvements. And the most crucial hyperparameter was the pair of activation functions, right? So it's where we can also use dedicated resources to study a specific group of activation functions for a specific trait and crop. So further studies will be conducted to increase the number of neurons in the second hidden layer and increase the number of hidden layers as well. Probably if we add two or three more hidden layers, we can have better results. And finally, we are trying to develop a sparse algorithm design in a way that we can find an optimal set of parameters in an easy way. So to obtain this result, we have to run the model 5 million times. So probably if we develop a system, right, that we can simulate, I don't know, 1,000 combinations, we can create a surface response or something like that in a way that can help us to reach the goal faster. Okay, now I'm gonna switch gears. I'm gonna talk about a different topic. And this is sparse tested design, how we can implement this to the industrial level. So as we know, breeders are interested in developing genotypes that perform other genotypes in a wide range of environmental conditions. It means that they have to conduct multi-environmental trials. And as we know, it's impossible, infeasible, or very difficult to test all genotypes everywhere. But probably because we have a budget restriction, or we just don't have enough seed to observe everything everywhere. Okay, let's review very quickly the hybrid pipeline and the industrial level. Initially, we have something like 150,000 genotypes that are observed in 10 environments, and then they select the most promising, and they increase the number of environments, and they keep doing this for several years, right? In a way, if we are lucky or the breeders are lucky, at the end of the breeding pipeline, they will release between 3, and if they are very lucky, to 5 commercial varieties. And this is time consuming. So the interesting part of this breeding pipeline is that the genetics that we had at the beginning is exactly the same at the end. So probably if we develop a method that could help us to identify the top performance genotypes at very early stages, we can save a lot of time. But we know the ideal scenario is impossible. Assuming that the environmental conditions won't change from the beginning to the end, and let's say that our target population of the environment is 100, probably the most convenient strategy will be observe the 150,000 genotypes in 100 environments. We know that it's not possible. We know because expensive, we don't have enough water, land, seed. But let's analyze what is happening along the breeding pipeline. So we had in year 1, they observed 150,000 genotypes in 10 environments, 1.5 million combinations. The second year, 10,000 in 20 environments, 200,000. So if we go to the bottom, at the end they observed something like 1,760,000 phenotypes, 50,000 phenotypes. So this is around 12% of the total number of combinations, assuming that the environmental conditions won't change from one year to another. As I said before, a more convenient allocation design will allow us to evaluate, I'm not saying to observe, to evaluate the 150,000 genotypes in the 100 environments. So we already are investing resources to observe 12%. So probably we can use some of these resources to evaluate a portion of the genotypes in as much environments as we can and predict the remaining, using that information. The target population of the environments is 100. At least we have to be able to survey the 100 environments with a few genotypes. So the objectives of this study were to assess the convenience of implementing these sparse testing designs to reduce the number of years for selecting these outperforming genotypes. We evaluate different methods to select training and testing. And we have preliminary results that we already published in other studies, where we were considering different combinations to compose the training set. What happened when you have the same set of genotypes observed across environments, or when you observe each one of the genotypes somewhere in any of the environments. And the motivation of this study was because I was interviewing for a research center in Philippines, and they asked me, what's more convenient, to have same genotypes observed across all environments or observe more environments? And I didn't know. I thought, my feeling is that it's better if we observe more environments, or even if we observe less genotypes, because we can make up the performance of the unobserved combinations using genomic selection. And this is how we started the design of this study. So we tested multiple different combinations, right? Same genotypes across environments, or genotypes observed only once in any of the environments. And then we saw that things vary depending on the crop. Also, we tested different ways, to compose the training set. So we varied the genetic diversity, and then we found things like we can improve predictability by twofold compared with conventional genomic selection. So in this plot, we have in the left side the case where each genotype is observed only once in any of the environments. In the right side, a common set of genotypes observed across all of the environments. And we observed that there are not that significant differences. So many people think that if they observe the same genotypes across all environments, they will have a better signal. But again, it has the problem that usually we don't have enough seed. So with this study, we show that it doesn't matter, right? So we consider 3 models: a model with main effect of the markers in gray, GY interaction, and a model that in blue, and a model that includes interaction with families. So this was for a soybean study. And now we switch the idea of the sparse testing, that we have very few genotypes, 1,000 genotypes in very few environments, 9 environments. To this case, we have a large data set from a company, soybean data set of 2,500 genotypes observed in 340 environments, all genotypes in all environments, for a total number of 850,000 yield records. So they have also genomic information on 2,300 markers, and we consider different prediction strategies, right? We said, what happened if we just observe a sample, a reduced percentage of all these combinations, and we use the results of the phenotypic data to predict the remaining combinations. So we vary the training set size between 0.3% that corresponds to 2,500 genotypes only, phenotypes, to 2% that corresponds to 17,000 phenotypic records, and we predict the remaining 833,000. So for selecting the 2%, we consider different strategies. So one strategy considered to select 50 genotypes in a way that we observe 50 genotypes for each one of the environments. 7 genotypes were unique for any of the environments, and there were 43 genotypes that were common with the neighbor environment. And the second allocation scheme corresponds to select randomly 50 genotypes. And we observe the 50 genotypes across all of the environments. Obviously, we have to conduct replicates in order to avoid some specific bias. So we took 2 replicates of the first method, 2 replicates of the second method, and also we implemented a method that we call supersaturated design that allows to increase or decrease the genetic diversity for a given set of genotypes. And in extremis, we select genotypes in a way that we observe 7 or 8 genotypes for each one of the environments. We were curious what happened if we reduce the genotypes in a way that we observe each genotype only once across environments. So these are the results. So for the first case, where we select genotypes, 50 genotypes per environment in a way that 7 are unique and 43 are common with the neighbor environment, for the 2 reps, we have in the x-axis the result for main effect model, y-axis the results for G-value model. Each point corresponds, or each dot corresponds to the correlation between predicted and observed values with both methods. So the vertical line, the horizontal orange line corresponds to the perfect match, and whatever dot is above the line represents that the G-value model was better. So in this case, for both replicates, we had the G-block model return a correlation of 0.236, while the G-value model 0.57. So we double the predictability. So remember, we are using 70,000 data points to predict the remaining 833,000. So I don't think the results are bad using only 2%. Probably the optimal might be using 5%. We can select the genotypes in 1 year, pretty much. So for the second method, where we select randomly 50 genotypes to be observed across all environments, results improved. So we have correlation with 0.294 with G-block model and almost 0.6 with G-value model. We keep improving. We were a little surprised because we are observing just 50 genotypes. Again, it might be the case we don't have a snot seed, so the results were not that different than the previous one, right? And finally, we consider the method to select a sample that maximize or minimize genetic diversity. This is the PC1, obviously. The label is grown there. We are decreasing genetic diversity. In the right side, we are increasing genetic diversity. And we tested those 50 genotypes, and we tested what happened predicting the unobserved combinations. And we have results when we increase genetic diversity and decrease genetic diversity. And we found that there are no significant differences. So G-block model reduced a little bit predictability to 0.232. Reaction norm 0.57 when we increase genetic diversity, and decreasing genetic diversity, results were the same. So there is a point where we have so many genotypes that probably it won't matter the training composition, right? And this is what we obtained. And finally, using 0.3%, only 2,500 genotypes as expected, results drop. So we have G-block return 0.2 versus 0.27. We knew that something like this would happen. It's just 0.03% to predict the remaining 99.7. Anyway, still the reaction norm model was better. So we found with this that the identification of superior cultivars can be reduced in time, right? If we implement this type of method, we can save a lot of resources, but the most important resource is time. No need to increase budget for already large-scale programs. So we saw in the industrial pipeline, they are already investing around 12% of all the resources to observe all the potential combinations, and here we use only 2%. Also, we found that for a given budget, we can increase the testing capacity easily by 9-fold. Or for a given target of genotypes and environments, we can reduce the phenotyping cost by 90%. Okay, that's about the first two topics. Now I'm going to talk about CHiDO. And what is CHiDO? So as I said before, it's cool in Spanish, but for us, CHiDO is a no-code application that aims to remove technical and financial barriers associated with modeling genotype and environment interactions in a prediction context. So the idea was taken from trying to help emerging breeding programs. I know University of Florida, Texas A&M, Wisconsin, you have a large number of resources compared with emerging breeding programs around the world. Right? You can hire a statistician, a quantitative geneticist, somebody good for programming. I mean, Corteva has like 3 different teams. Every time I met them, like using somebody new. But emergent programs, they cannot do it, right? So I have developed many pipelines for CIMID, for example, or for other collaborators. And the idea was to craft something that anybody can use or can access around the world. So last year, this CHiDO stands for Characterization and Integration of Driving Omics. Last year, I was lucky that a student contacted me. And this guy, Francisco Gonzalez, he was a master student with me. He was a student at night, and during the day, he's a model apps lead for Google Cloud North America. So he might know something, or a little bit about programming. So it's when I try to take advantage of that. Also, I had the visit of Julian Garcia Vadillo, a PhD student, and later on he was intern at Google too. And now I have another visiting student, Victor Sage. He's a PhD student from University of Viçosa. So this team helped me to craft, right, the prediction pipelines that I developed for other collaborations in a software that does not require coding. That is CHiDO. So the main characteristic now of CHiDO is it allows the integration of omics in a very easy way. So we might have genotype level omic. So we have in this toy example 3 genotypes or genomic information for 3 genotypes, and we can compose a covariance structure with that. So yesterday during the workshop, we were mentioning a little bit of how we use a trick of converting metrics of data in a covariance structure, right? We just multiply by its transpose, and then we have a kernel. And we can have omics for different levels. We can have omics at the plant level. For example, when you are flying the drones, you can collect high throughput phenotyping data that the genomic information from one, the information you are collecting from one genotype in one environment is going to be different in a different environment. In a way that if we have replicates, we have to use the design metrics to redistribute that covariance structure across all genotypes in all environments. And we have an extended genomic kernel, and we multiply by the kernel at the individual level. And then we have an interaction. So it's very easy to include interactions in this way. And also we evaluate the variance components. And this measure is important because we can identify what are the key factors driving variability. So we have different omics, we have different assumptions. So we assume normality for easy, and then we evaluate the contribution of the variance components to explain phenotypic variability, and we can play around. So you can identify something like this. It looks a little bit like the heritability, but we can test pretty much what's the contribution for all of the different omics, right? Like a percentage of variability explained, but more important, we evaluate trait performance based on 4 different cross-validation schemes. CV1, where we are predicting new genotypes that we didn't observe across environments, but we observe other genotypes in those environments. CV2, the case of predicting incomplete field trials, where some genotypes are observed somewhere, but not in other environments. CV0, predicting new environments, so we never observe the environment, and we are just trying to guess based on the means with the other environments. And CV00, where we are predicting new genotypes that we didn't observe in environments that we have not observed yet. So CHiDO is an R-Shiny app that implements the concepts that we are showing in this presentation, checks data integrity, and connects the different omics. So that's the power, right? We add the different omics, and it starts aligning. Builds covariance structures, so once we compute the covariance structures, we can provide these as arguments in the model. Generate cross-validation schemes. For now, we have annual crops, but new release is going to include, for sure, perennial crops. And we compute the performance metrics, so variance components and correlations and mean square error. So currently, we can conduct forecasts of trait performance, hybrid prediction using general combinability and specific combinability in interaction with environments, so environmental covariates. And the next release will allow GBI analysis for stability studies, sparse testing design, deep learning, et cetera, et cetera. So how can I have CHiDO? So we can use CHiDO on demand, right? We have a website where you can go there, upload your data, and play with the implementation. And this is how CHiDO look like. Or you can download the implementation in your local computer. The advantage of doing that is that you can, running analysis online, we have some restrictions about the number of resources that you can use. We have limited resources. If you do this in your computer, the limit is your computer pretty much. So you can go to this website, the GitHub account, you can clone the repository, and then you can run this through R. And this is the interface. So we have an interface to upload the omics, another to create the models. This is my favorite part because it seems like we can create models like playing video games. And then we choose the different cross-validation schemes that we want to analyze, and then we view the results. So I'm going to show you a little bit of this. So we upload the data, so we can pick whether we want to run conventional genomic selection models or whether we can use general combinability or specific combinability terms. And then we select the data types. So we can use genomic markers, and we have to pick or introduce the information from the phenotypic information, which column has the ID of the genotypes, right? The name of the genotypes, the name of the environment, and which column has the trait that we want to predict. So, and this is convenient because if you have multiple traits, you can just change that input there. Or if you have data that is taken at the plot level, you have a compound ID that you can also introduce. And then you have environmental markers, so high throughput data, and then there you have to specify which column is going to help you to align with the metrics of phenotypes. So we can compose the linear predictors in a drag and drop way. So you have the terms that you can add in the model in the left side. You just drag and drop to the right side. And if you want to introduce interactions, you drag and drop 2 terms or 3 terms or whatever number of terms. You let them to collapse. Or you can also type the model in the box. So you don't want to play video games, you can just go and type the model directly. And you can name the model the way you want. Later on, we have to train and validate. So this is a Bayesian approximation. We have to pick the hyperparameters as number of iterations. We recommend something about 20,000 and burning rate, right? Probably 5,000 will be more than enough, and a thinning of 5. No, not thinning here. So we have the number of, for the cross-validation, number of faults that we want to use. And if we want to apply quality controls on the data, right? If we want to eliminate those markers with more than 20%, we specify those things here. We center and standardize the inputs because we want to, or we might want to compare, right, the contribution of the different omics with respect to the phenotype. So we have to center everything on one. And then we pick the different cross-validations that we want to use. It could be CV1, CV2, or all of them. And then we have, for each model, 6 steps, right? That it can help us to track the time for finishing. So prepare the incidence metrics for each one of the components, compute the covariance structures, et cetera, run the predictions, and then you will get the metrics. So I'm going to explain very quickly how this works for hybrid prediction. So we know the breeding programs can focus on development of hybrids to test lots of single cross combinations between lives that belong to different heterotic groups. So this might be time consuming, and because we might have a very large number of potential combinations, right? We cannot observe everything everywhere, but we might have samples, right, of these hybrids observed in some locations. For example, I'm sure all of you are familiar with genomics to fields, right? We have sets of hybrids observed all across the US. Obviously, they reduce GBA interaction by selecting locally for the different locations. That makes sense. But in those cases, we have genomic information of the parents, right? And we can use that to make up the genetics of the individuals. And this is how the input look like. So we have a matrix of phenotypes where we have a column for environments, a column for the name of the hybrids, the trait that we want to analyze, and also we specify what are the parent 1 and parent 2. If we have missing values, we have to specify those as NA. And then here we pick the combining ability. So there, we are going to add that information, which genotype, which column has the genotype ID, which column we're going to use as parent 1, in this case, column number 4, which column is going to be parent 2, column number 5, and whether we have a compound ID. For example, if somebody flew a drone, so we can introduce that information there. And the target trait, in this case, is column number 3, right? Yield performance, probably. And then we add the omics. So for example, we have the genomic information of the parents. We specify which column, right, has the ID of the parents that we are going to link either with parent 1 or parent 2. And then we add other type of omics too. So we have to specify all the time what will be that column that is going to help us to link phenotypes with any of the omics that we are going to introduce. And then we assemble the model. So in this case, we have terms like environment, line of hybrid name, line 1, line 2, so parent 1, parent 2 with the marker information, or G1, G2 when we have marker information of the inbreds. And then we can collapse two terms, G1, G2, to create the specific combinability term. And we can keep building on top of this. We can add the interaction with environments, for example. And then we train and validate the hyperparameters. Obviously, we recommend something like above 20,000 iterations, and for burning, 5,000. We pick the different cross-validation schemes, and also we can fix the seed if we want to replicate results and compare with colleagues. And then when we fit the model, we have to fit one model at a time. So we pick the model and what are the cross-validation schemes that we want to analyze. And finally, we have the metrics. So one of the metrics is the correlation between predicted and observed values for each one of the environments, or we can have a weighted average at the end, or the mean square error, or the variance component. So all of these metrics will help us to take a decision. Probably we can identify what is the factor that's driving variability easier in this way. Okay, now I'm going to talk very quickly about another type of analysis that we are implementing in a new release. and it has to do with stability parameters. We know there are many methods, and one of the most conventional methods is AMI analysis. This is a very great tool to visualize interactions in biplots. However, the downside is that we cannot attach uncertainty parameters around the biplot. So a few years ago, we proposed the first Bayesian approach, where we were able to draw these. Bayesian doesn't like when we say confidence intervals, but high posterior densities that act as a confidence intervals. And then other authors later on developed other metrics in the frequencies approach, and later on they use the Bayesian approach to enrich the results. We have some frequencies approach like WAS and WASB index, where they take into account the performance and the stability, and more recently, stability index based on the Mahalanobis distance. It's a paper that is going to be submitted soon. And this is the input data. So we have a column for the site and to replicate, and the observed values. We tune the hyperparameters. Here we recommend something above 30,000 iterations and 5,000 for burning, and a tin rate of 5. And then we pick what are the different metrics that we want to analyze. By default, we have to always use the AMI model, because the results of the Markov chain are going to be used to create the other stability parameters. Also, we can combine stability with performance, because we might have a genotype that's very stable, but the performance is bad. So we would like to balance that. We can say, okay, 90% probably is stability, 10% performance. So we can pick that here too. And these are the results. We can visualize the trace to evaluate whether we have convergence or not. We have the Bayesian biplot. In this case, we don't have the contours for now, but we can use this to create or to build up the other parameters. For example, WAS index, compute stability of genotypes. The lowest values correspond to the more stable genotypes. Again, but it might be the case that the performance is not that good. So we can use WASB is a modification of this index, where we take into consideration yield performance, and in this case, higher values are better. Or we have other methods like STAPDIS, where we can identify quadrants, right? The genotypes will be in quadrants. In this case, those genotypes in quadrant number 4 are the most stable with higher performance and the narrow variability. So we can find those things. So what's next for CHiDO? As we saw, it's a powerful tool to integrate and conduct estimations and predictions. Its implementation is not exclusively of breeding applications. We can apply this to other areas like personalized medicine, interaction with pathogens, optimization for the allocation of resources, integrate AI methods using the next stage, and the structural equations to find causal relationships. So the main goal is to develop an open source suite of applications to ease the data integration and analysis of small and emergent breeding programs. So we are open to suggestions, ideas, partnerships, etc. So sorry for this self-promotion, but you don't have anything to do during the summer. You are more than welcome to attend to the third short course that we are organizing in beautiful Gainesville from July 7 to 11. So the previous 2 years, we were very successful. We have over 90 attendees coming from 12 countries, 5 continents, 26 states in the US. And this year we are partnering with Driscoll, University of Georgia, University of Arkansas, Agronomics, Colegio de Postgraduados, Polytechnic University of Madrid, NVIDIA, and the UFIT Research Computing, and more speakers. So you are more than welcome to attend. So with this, thank you, and of course, go Gators. So I'm more than happy to take any of your questions.
Thank you, Dr. Jarquin. We have about 8 to 10 minutes for the Q&A. Questions?
Thank you for the nice talk. So I guess my question is a bit about for people who work on outbred crops. I mean, I see you have a lot of nice tools for handling the complexity of G by E, but what about the complexity of the G part of it? Is that something that you're thinking about incorporating into CHiDO or some other tools?
I mean, we have not thought because nobody asked the question, but that is a very good opportunity for me to come here and explain what we can do with CHiDO, because that might be something that we might consider to integrate in the new version. So thank you for the suggestion.
Questions? Okay, I'll have one question. Like, since for the deep learning or like for other models than zBlob, like, do you see like value in using that, or like, what are the scenarios like we can use a non-linear models other than zBlob? But like, based on like what I run, like in most of the case, like it did not improve that much. So what is your take on that?
I mean, it depends on how much you can influence the trait, because, I mean, a breeder is going to tell you that we know that parents pass alleles, they don't pass genotypes. So if we use more elaborated models, we are pretty much implying that we can control the way how these alleles or these genotypes are being passed through the generations. Now, it depends on the ultimate goal. If you are a breeder, you care about genetic gain. If you work for a company that's going to sell seeds, it doesn't matter. You can add all of the information that you can have at hand, because your ultimate goal is to be very precise predicting amount of yield that you're going to have to sell. In that case, whether it's driving variability, it doesn't matter. You already have the product. You just want to know how much you can sell. So it depends. It's always something I teach during the class. Like, it depends on your goal. You can use one or another tool, right?
If we don't have more questions,
Thanks, Diego. Can you talk more about the omic integration? You know, with genomic data, you usually have biallelic data. The omic data is quantitative usually. You didn't show much in terms of phenomic features that you might have included in these. And then are those going into a GBLUP type approach, or can they also be used in some type of like ridge regression or anything like that?
I mean, if you check in our GitHub, we have exemplary data where we use the integration of high true phenotyping data. So data was collected with drones. And for now, we are just relying on covariance structures because it can reduce the computational burden. It's equivalent to RBLOB, for example. And we might be interested later on probably to test other type of models. But for now, we don't want to complicate things that much. Thank you.
Diego, thanks for the great talk. I was wondering if you would be willing to just lay out for us what you anticipate the benefits would be of adding more neurons to the second layer and or adding more layers beyond the second.
Okay. Yeah, I mean, we are on a test. What happens if we are adding, I don't know, 3, 4, 5, 6, 7 more hidden layers? So this was kind of for a master thesis. And then we're going to be able to survey what will happen if we have more neurons or other type of architectures, especially because we want to develop first a method to optimize. Something that has not to be that exhaustive like this. But it was a very first step, I would say. Now we are going to build on top of this to see if we can optimize the selection of the parameters in an easier way. Thank you.
If we don't have more questions, let's thank Dr. Jarquin for his talk. Thank you.